internship with ELIXIR.
Sorry,
and
I didn't ask you, Ellen, if you're okay with recording. I hope it's okay.
Yeah, that's fine with me. Thanks.
Thank you.
So today we are very happy to host Ellen and Zhong. So Ellen graduated from the University of Virginia in chemical engineering. Then she worked for some time with DSHO as scientific programmer, where she worked on new methods to predict small molecule to protein binding affinities using molecular dynamics simulations. Then she started a PhD at MIT, where she worked on at the interface between machine learning and cryo-EM and structural biology, which is particularly of interest for us since we have we have a lot of research in cryo-EM going on, and development of new technologies related to time-resolved cryo-EM. So Ellen also spent research summer internship at DeepMind, and now she just recently graduated. So as you maybe overheard our conversations, she's now deciding on academic position, academic research, or joining DeepMind. So Ellen, I'm very happy to have you today. So I'm going to stop sharing the screen and let you take the lead.
I just ask everybody to post the question on the chat, and we'll have a discussion period at the end of the talk, and that's a very important part of the webinar series. So please do ask your questions, and welcome, Ellen. Go ahead.
Sure. So how about I kind of target 40 minutes, and then we'll have plenty of time at the end for questions.
Wonderful.
Okay, excellent. Thank you again for the invitation, and also thank you for that kind introduction. I'm very happy to be here, and it's nice to be able to, now that Zoom is here, to be able to share with my work with people across the Atlantic Ocean. So I'll be talking about CryoDRGN, which is a machine learning method for heterogeneous cryo-EM reconstruction. And just to overview some of the papers that describe this method. So this first appeared at ELIXIR, which is the machine learning conference. And this was joint work with Tristan Beppler, who is a former graduate student in Bonnie Berger's lab, and then my two co-advisors, Joey Davis and Bonnie Berger. So later appeared when we extended the method to real data sets and kind of showcased its ability to kind of uncover dynamics. And this appeared in Nature Methods last year. And then very briefly at the end of this talk, I wanted to mention two kind of follow-up extensions, which is how do we analyze CryoDRGN models to really look at the distribution or kind of the structural landscape. And then the second is ab initio reconstruction, so extending the capabilities of cryo-EM reconstruction. And this is joint work with Ashwin Narayan, also a graduate student in Bonnie's lab, and Adam Lair at Facebook AI, or Meta AI Research. So I probably don't need to go into the motivation since we're at a structural biology center, but as we all know, all essential biological processes are carried out by proteins and protein complexes. And so one of the goals of structural biology is to really understand the function and the mechanism of these macromolecular machines via their three-dimensional shape or structure. And what these illustrations from David Goodsell are really beautiful. They really show the diversity and the shapes and sizes of these macromolecular machines, but they are static. And so we also know that in order to kind of carry out their functions in these fundamental processes of life, these protein complexes are inherently dynamic. They have to change conformations and or composition in order to carry out whatever biological process. So for example, here we have the spliceosome splicing cycle, and you can see there, this is just a schematic. And so clearly there's a lot of things going on. And then just as a, just as a teaser, this is a CryoDRGN reconstruction of the pre-catalytic spliceosome. So unfortunately, techniques to study these molecular motions, especially at atomic level resolution, are rather limited. So we have NMR, which is limited to rather small proteins, less than 100 amino acid residues in length or shorter. Then there's what I like to call kind of avant-garde crystallography-based approaches. This is X-fill crystallography, which can look at some of the mechanical deformations within these protein crystals, but this still requires simple crystallization. And so that might be a bottleneck in terms of what complexes you can study. Then my background, my previous background before getting into cryo-EM, which is in molecular dynamics simulations. So what I'm showing here on the right is an MD simulation of the SARS-CoV-2 spike protein. And while this gives you a very kind of precise in silico microscope at what's going on, there's a lot of challenges associated with the accuracy and also the compute cost, the accuracy of the models and also the compute cost of running these simulations. And so that's why I've been very excited about the potential of cryo-electron microscopy or cryo-EM at studying kind of the ensembles and dynamics of these macromolecular machines. So just a very quick overview of the cryo-EM image formation model. In single particle cryo-EM, we have a purified solution of our sample of interest that's fixed in a very thin layer of ice. And then imaged with the transmission electron microscope. So we have these particles here that are randomly oriented before being flash frozen and then projected. So unlike this kind of idealized projection image shown here, cryo images are typically very noisy due to the low electron dose required to limit radiation damage. So that's kind of the forward image formation model. The goal of reconstruction, which is the focus of my work, is to solve the inverse problem. So how do we go from a data set of single particle cryo-EM images to the 3D cryo-EM density of our protein of interest? So there's typically hundreds of thousands. These days, it's easily millions of single particle images, and we want to reconstruct the 3D structure. So there are a number of challenges associated with this task. First and foremost, we don't know the orientation or the pose of each of these particles. Very noisy, like I just mentioned. You have to take into account the contrast transfer function or these image degrading filters in microscopy, as well as discretization of the measurements. And then there's kind of the so-called heterogeneity problem. And so this is where we break the assumption that each of these images captures an identical static structure. In reality, each of these images is a unique particle. And so if we could reconstruct the distribution of conformations, then we could study kind of the ensemble structures of these macromolecular machines. So I just mentioned each image contains a unique molecule. And so this is actually a major opportunity in single particle cryo-EM, unlike in crystallography, where each copy of our complex is rigidified into the exact same structure in our crystalline lattice. Here we actually we collect the data to study the ensemble of structures. But you can imagine how this complicates reconstruction. And so there are several kind of heterogeneous reconstruction algorithms now. The kind of standard approach, other than discarding as much of your heterogeneous data as possible, is something called multi-class reconstruction or 3D classification. And so this models the heterogeneity of our complex as a discrete mixture model of K independent structures. So this would work well if you have maybe a couple of discrete or distinct states in your system, but you have to pre-specify kind of how many states you have. And in general, this would kind of fail for maybe more complex forms of heterogeneity, which you don't know a priori. And just to give a typical cryo-EM processing workflow example, you can find this in the supplemental information of a lot of cryo-EM structure papers, where at the top you have 3.7 million images in your pick particles in your data set. And then through many rounds of this multi-class reconstruction approach, at the end yields 2 structures, and you've also thrown away a significant amount of your data. And so this kind of discrete 3D classification model in particular is limited for modeling continuous forms of heterogeneity. And so one of the goals going into this project is, can we come up with a way for reconstructing continuous distributions of protein structure from single particle cryo-EM data? So our approach is called CryoDRGN. CryoDRGN is an acronym. It stands for Deep Reconstructing Generative Networks, and it frames heterogeneous cryo-EM reconstruction as unsupervised learning of a deep generative model of 3D structure that's trained on the 2D kind of single particle cryo-EM images. And so there's a couple of aspects of this method that I won't go into too much depth, but just briefly to summarize, the first is a specific neural network architecture for modeling 3D structure. Our overall kind of machine learning system is an image encoder volume decoder architecture that's based on the VAE, or the variational autoencoder. And then the way we train the system is with exact inference for poses, or those camera unknown orientations, and variational inference in the style of the VAE for heterogeneity. Okay, so going into kind of those aspects in a little bit more detail, the key idea in CryoDRGN is that we use a neural network to model the 3D density map. So instead of representing the structure as kind of on this discrete three-dimensional voxel array, we instead train a neural network to approximate this continuous 3D function from 3D Cartesian coordinates to the cryo-EM density at the location. So CryoDRGN structures are parameterized as a neural network instead of a voxel array. So the goal of reconstruction in this traditional setting is to kind of fit the values of the density at each of these voxels. In CryoDRGN, we want to learn the weights of the neural network to best approximate the continuous 3D cryo-EM density function. There's a kind of architectural or technical detail, which is that instead of directly inputting the 3D Cartesian coordinates, we featurize it using positional encoding function inspired from the transformer literature, consisting of sinusoids of different frequency. So this featurizes the input to help with kind of optimization of the neural network. And this particular architecture for modeling kind of 3D functions it's been really exciting to see that this has completely taken off in other computer vision techniques, other domains of computer vision on natural scenes. So if you're interested in kind of learning more about that, you can check out NERF. So why do we use this neural representation of structure? The primary reason is to model heterogeneity. So in this discrete 3D classification approach, or multi-class refinement, which I call here, the goal of reconstruction is to fit kind of K separate voxel arrays. Typically you have to specify K, you also have to specify initial volumes, which is a significant source of bias in this 3D classification approach. And typically K is relatively small, so less than 10. In CryoDRGN, in contrast, we extend, so this neural representation gives us this way of really of extending the input dimension of our neural network to include this latent variable, which is a very generic way of describing heterogeneity. So this latent variable vector, what is this latent variable? So I guess to give some intuition, if our latent variable were one-dimensional, you can think of this as position along some reaction coordinate. And in general, this gives us just additional degrees of freedom for modeling heterogeneous structure. Maybe you can think of it in a slightly higher than one-dimensional case. You can think of Z, or latent variable, as denoting position on some free energy landscape or conformational kind of landscape. So that's CryoDRGN's generative model, is we're modeling kind of structures as from this continuous distribution. This encodes an n-dimensional continuous distribution over structures. And the question now for reconstruction is, how do we learn this model from data, from our kind of 2D single particle cryo-EM images? So the overall architecture has this image encoder volume decoder architecture. In single particle cryo-EM, we know that images are slices out of the 3D volume in Fourier space. So this gives us a very natural way of kind of relating the 2D images as slices out of our 3D volume using our coordinate-based architecture. So kind of describing how we train the system, given an input image, our encoder, which is just a simple MLP, maps the image to kind of to the latent space. So we have some latent code or latent representation of this particular particle. Then the volume decoder, which is that coordinate-based representation that I just described, takes a sample or takes the latent code, and then the kind of 3D location of the image. So we have the slices, and then reconstructs that image or the slice pixel by pixel. Then we can compare the reconstructed slice to the input image, compute the loss or compute the error, and then use that to update the weights of the neural networks. And so the training objective, so this is, so the neural networks are randomly initialized, and it's trained kind of on a single particle cryo-EM data set to maximize the likelihood of the data. So that's the training of CryoDRGN. To evaluate it, so now once we've trained the model, we have these two neural networks. We can take our single particle cryo-EM data set and encode the images into latent space. And then we have this latent space representation, or kind of this latent embedding of each image. So instead of a full n by n image, now we have this low dimensional vector representing each image. And we can just view that distribution or view this latent space representation of our data set. And maybe this already tells us something interesting about kind of the heterogeneity of our system. But we can also use this decoder neural network to generate volumes at different values of our latent variable. So maybe there's this cluster out here that we're interested in or want to investigate. We can decode an entire three-dimensional density map given that location in latent space. And we can view representative samples from all across our latent space representations. Since this is a continuous model, we can also generate continuous trajectories. So if there's two end states of interest, we can, if there's two end states of interest in our latent representation, we can kind of interpolate between those two end states and then decode structures to get a continuous trajectory. If you don't want to, or you want to kind of validate this neural representation, you can always take the underlying images and then do a homogeneous reconstruction with traditional tools. So that's kind of shown here, where we're taking some subset of our particles and then performing validation with traditional tools. Or perhaps if you just look at, oftentimes there's a lot of junk particles in real single particle cryo-EM data sets, so you can just filter the data set based on the latent embeddings. So just to summarize, given an imaging data set, two neural networks are jointly optimized to learn the encoding and decoding of this latent space describing the data set's heterogeneity. And there's two training regimes. So the first is ab initio heterogeneous reconstruction, where we don't know the poses. We don't actually know the orientation of those slices, and we are jointly fitting that. And I'll describe that later at the end. The second is heterogeneous reconstruction with consensus poses. So this is the version that's described in our Nature Methods manuscript and is publicly available on our GitHub. And here, what you're doing is you have to run an upstream homogeneous reconstruction in your system to assign all the poses of the images and then fit the residual heterogeneity of the in the data set. So just in terms of compute requirements, it's very similar to other single particle or reconstruction software. So you'll need a GPU workstation. This is all implemented in PyTorch, which is a very kind of standard deep learning library. And then in terms of the input data requirements, typical single particle cryo-EM images with the high quality pose estimates from our consensus reconstruction, as well as accurate CTF parameters. I won't go into kind of the software, but ideally, we've written this to be relatively easy to use, relatively straightforward, and there's also a very extensive tutorial now available. So next, I just wanted to go through a couple of examples of applying CryoDRGN on real data. So the first is applying CryoDRGN to fit residual heterogeneity in kind of original homogeneous data sets, or high resolution homogeneous data sets. So this first test was, can this neural network model for structure, can this neural model learn high resolution cryo-EM density maps? So here we've trained the CryoDRGN decoder without any latent variable input on images from this Plasmodium falciparum ADS ribosome dataset. And on the left, we have the reconstruction from CryoDRGN, and on the right, we have the corresponding voxel-based reconstruction, I believe, from CryoSpark. So you can see we can learn, both methods can kind of resolve high resolution details of this ribosome complex. Just to give some intuition there, the specific architecture of our neural model kind of bounds the attainable resolution. So I kind of think of this similar to the box size that you use in reconstruction. With a larger neural network architecture, you have more parameters, so you can train or you can learn high resolution details, but it also trains slower. So there's some inverse trade-off there. And we've also done a lot of work kind of exploring where exactly is the, or just to establish best defaults, best practices there as defaults. This particular data set of the ADS ribosome has become a benchmark for various other methods for heterogeneity analysis. So I wanted to mention a couple of others, although in the last few years, there have now been several other methods. So multi-body models heterogeneity as originating from separate rigid bodies. And so based on these user-defined rigid bodies, which are allowed to move relative to one another during rotation, during reconstruction. And then we have CryoSpark's 3D-VA, which fits this linear subspace model. And so this gives us a sense of the overall heterogeneity, but the actual model is a linear model. And so any kind of motion will be expressed as linear interpolations. So there's oftentimes these artifacts from linear interpolation. Here's where we get when we train a CryoDRGN model in this particular data set. So what I'm showing on the left are the embeddings of the images. And this was originally a 10-dimensional latent variable model, and it's projected into 2D just for visualization purposes here with PCA. And here are 20 sampled structures, kind of denoted here. One of these structures here, 13, is actually the rotation of the small subunit relative to the large. But in addition to that, we can see kind of heterogeneity in a lot of the other elements in both the large and the small subunit. And just to compare those two in a little bit more detail, here we can kind of see this rotation between the blue and the purple, two density maps, as well as the concomitant kind of motion of this RNA stalk, and then the disappearance of some of these RNA and protein helices. There's also, so a lot of people have asked, how well does CryoDRGN work on smaller protein complexes? So just another quick example, this is the RAG1, RAG2 complex, which is, I think, around 300 or 400 kilodalton. And again, we can learn kind of the high resolution homogeneous structure in CryoDRGN. When we train the model in the heterogeneous setting, we can see that there's actually a lot of heterogeneity in these kind of asymmetric DNA elements and some of the protein subunits down here. So that was very exciting to see. So those were data sets that were originally kind of, the original published structure was a homogeneous reconstruction. Next, we wanted to explore how CryoDRGN would work on a very heterogeneous data set. So this is the assembling ribosome. So originally, this data set had 131,000 cryo-EM images containing a mixture of assembly intermediates. And so this was done by my advisor, Joey, during his postdoctoral work, and through many rounds of like user-driven classification, they were able to identify 4 major states labeled B, C, D, and E. And then within those major states, performing more classification to identify these minor states, yielding a total of 13 minor states of the LSU, or large ribosomal subunit. So what happens when we train a CryoDRGN model in the system? So what I'm showing here are the, again, latent space embeddings of the single particle cryo-EM images. They were 10-dimensional again, and we're projecting it or visualizing here in 2D with PCA or with UMAP, which is a nonlinear kind of dimensionality reduction technique. We can already see clusters in the latent representation. Comparing those clusters and our latent space representation with the 3D public, we can compare these clusters to the 3D classification labels, and we can see that these clusters correspond to the major states of the LSU. So each of these single particle images are basically coloring by the published class assignment. But we're not just limited to our latent representation, we can also generate the structures. So generating the density maps at the centers of these different clusters, we can see we get the kind of 4 major states of the large ribosomal subunit. So assembly state B is the least mature. C, we have the base of the large ribosomal subunit. In D, there's the central protuberance. And then class E is the most mature form. Just to show some additional examples. So class A was actually, the sign class A from the original publication was actually an impurity during sample preparation, which was the 70S ribosome. You can see that there were some particles that were mixed into this data set, and those were separated out very readily by CryoDRGN. And then showing some additional samples within the purple cluster for the minor assembly states. You can see that there's variation consistent with assembly states E1 through E5. We were also very excited to see, originally kind of confused or curious what this strip of purple particles were next to the green cluster. So what this means is that these images were originally classified into class E, or the mature assembly state. And when we generate a structure from this region of latent space, we got this new assembly state that resembled class C, in that only kind of the base of the ribosome or large ribosomal subunit existed, but there's this helix dock that was previously only associated with the mature form of the large ribosomal subunit. We can take the particles here, so there's around 1,000, a little bit more than 1,000 particle images, and perform a homogeneous refinement, and we get the same features. So this was all kind of generated from one training run, or I guess there's two training runs. The first, we did some filtering of the data set, but then this whole kind of ensemble of all the minor and major assembly states and this new assembly states were generated after training the data set once on the whole, training CryoDRGN on the data set. So that was this case with lots of kind of compositional heterogeneity. Next, we were interested in actually seeing using this tool to visualize continuous motions. So that was one of the original goals of this method. So here is we analyzed the data set of the pre-catalytic spliceosome. In the original publication by Ploshka, Linen, and Guy, there was one published structure, and because of the heterogeneity of this complex, the different subcomplexes were resolved through many rounds of focus classification and then kind of just put together to generate a composite map. This was also analyzed with multi-body refinement based on user-defined rigid bodies. They were able to see kind of motion of this top SF3B subcomplex, but because of this rigid body assumption, some of the interfaces between these rigid bodies contain artifacts like the breaking of this helix between this SF3B and the core. And just for example, this is CryoSpark 3D-VA. Again, because of the linear subspace model, we see the motion of this SF3B, but it manifests as this kind of linear interpolation instead of the true motion. So this is what we get when we train CryoDRGN on this particular data set. Here I'm kind of showing trajectories along the principal components of our latent embeddings. Along PC1, you can see the same large scale motion. Along principal component 2 of our latent embeddings, we see that the SF3B and helicase subcomplexes are moving in opposition. And so again, this gives us a very nice sense of the overall heterogeneity within the data set. One potential caveat is that the interpolation along the PCs can produce non-physical motions if we're interpolating along regions or through regions of the latent space where we don't have data. For example, if there's compositional heterogeneity. And so because of that, we're interested in kind of staying on the data manifold or staying kind of within the distribution of our latent embeddings. And so what I have here is this graph traversal algorithm where we're walking along the k-nearest neighbor graph of our latent embeddings, generating structures at the visited locations, and then putting them all together to form a trajectory. And so coming from an MD background, this is really cool to see. This really resembles maybe a plausible trajectory of this macromolecular machine in motion. So this gives us one way to kind of just visualize and explore the full complexity of the learned distribution. And so those were kind of analyzing previously published data sets with CryoDRGN. We've also learned a lot now applying CryoDRGN to new data sets or not the final published image stack. So kind of starting out, it was because we can model both these kind of continuous motions as well as compositional heterogeneity. One of the questions we're interested in is like, okay, we have this model that can model all these complex forms of heterogeneity. How do we actually analyze it? And so the original approach was to kind of provide a lot of interactive tools to facilitate analysis of all these diverse forms of heterogeneity. So in particular, we have a Jupyter notebook that allows for exploratory data analysis. So visualize these latent embeddings in UMAP or PCA, kind of interactively visualize the raw images, as well as interactively generate new density maps. And so just to give an example of that, especially in a lot of these data sets being processed, what we found is that there's a lot of kind of non-structural heterogeneity within the data set. So these ice kind of grayscale artifacts, as well as maybe these edge artifacts from recentering the box. And so this interactive kind of lasso tool lets you kind of interactively remove particles that may interfere with training the model. And so in this particular case, after removing those artifacts, we were able to resolve the underlying structural heterogeneity. Before that, the model was very preoccupied with maybe modeling some of these weird artifacts. And so this particular data set is the SARS-CoV-2 spike protein, courtesy of the Wiesler lab. Just a few data set and training details. There's 276,000 particles. This took 10 hours to train. And we can see, that I'm showing 20 sampled structures here in our graph traversal trajectory, where we can see kind of continuous dynamics of the RBD, or receptor binding domain, as it transitions from the open and closed states. So as we've analyzed more and more data sets, one thing we're interested in is moving towards more automated analyses of the ensemble of structures. And so this is, I'll motivate this by showcasing this data set from Sue Fei, who is a former postdoc in Bob Sauer's lab at MIT. She trained a CryoDRGN model on their data set of ClipXP. They were able to see, the original publication showed this intermediate and recognition complex. They were able to see kind of the continuous transition between those two. And this is CryoDRGN's latent variable representation of the data set. Originally, I told her to generate a lot of structures, so you can generate an arbitrary number of density maps. I think we generated either 50 or 100, and she annotated the different regions of the latent space based on kind of the observations of what the different structures resemble. And so one thing that we've been interested in is, can we automate this? Can we kind of can we do this analysis to be more reproducible and summarize the distribution of structures. So this has been in collaboration with Ashwin Narayan, just how can we gain insight from these high dimensional biological data sets. There's kind of two interpretations that we take for how we analyze the resulting ensemble. One is a continuous version. So can we think of continuous reaction coordinates that describe the ensemble? And so this is performing kind of PCA in the volumes, and then using those principal components as reaction coordinates for kind of visualizing the distribution of volumes. And then the second is a discrete clustering approach, where we cluster the ensemble volumes using an agglomerative clustering algorithm. And this has been successful for identifying rare states that were missed in both the original kind of 3D classification, but also in the user-driven exploration of the ensemble of structures. So I think in this particular case, we were able to see this substrate engaged state, which comprised 0.3% of the data set. And this is available in CryoDRGN 1.0. And then the last thing that I want to mention is recent work extending CryoDRGN to the ab initio reconstruction setting. So again, this is in collaboration with Adam Lehrer and my advisors, Joey Davis and Bonnie Berger. This appeared at ICCV, a computer vision conference last year. And we're interested in performing reconstruction without any labels on the single particle cryo-EM images. And we want it to work on real data sets. So revisiting this training architecture, we have this Fourier slice theorem that relates 2D images to slices out of the 3D model. And I kind of hand waved this or skipped over this, but when we reconstruct that slice, we're kind of already giving it the oriented 3D coordinates. But in the ab initio setting, we don't know the orientation of the slice. So we need some way to approximate the pose or the alignment of this particular particle. How do we do that? How do we obtain this rotation and also translation for the imperfect centering of the particle? I wanted to mention there's a couple of paradigms, different paradigms for doing this pose inference procedure. I won't go into too much detail. You can train another model to predict the pose. You can maybe directly do gradient descent. There's also been some work on distribution matching and kind of this like adversarial approach to estimate the volume skipping over pose inference. Exploring some of these ideas, we weren't able to reconstruct, we weren't able to learn the pose because we got stuck in these spurious local minima. So this is no longer proteins. Here we're just dealing with this toy hand data set. So instead of kind of these variational inference or gradient descent based approaches, we instead kind of use the same approach as in traditional reconstruction software, which is a hierarchical search over the space of possible poses. So this hierarchical search procedure starts with a coarse kind of gridding of possible alignments and then successively refines that. And we also employ frequency marching, which speeds up and also prevents overfitting of this procedure. And with this hierarchical search, we can now reconstruct the correct volume. So here again, this is just doing kind of homogeneous reconstruction in the setting for CryoDRGN. One of the main challenges for CryoDRGN in particular, though, is the computational expense of doing this post-search procedure. So that's kind of why we were not keen on doing this originally. In traditional voxel-based reconstruction methods, to compute a slice, you're just taking the average of the 8 spatially closest neighbors. But in CryoDRGN, to compute the slice or kind of any pixel in our slice, we need to evaluate a full neural network and do all these neural network passes. So CryoDRGN models are much more expensive to evaluate. And a lot of the techniques in CryoDRGN 2 are specifically tailored towards the neural model setting to make reconstruction or ab initio reconstruction, specifically pose search, computationally tractable. There are many hyperparameters to tweak. I won't really go into this, but after kind of doing all those hyperparameter searches, we were able to perform reconstruction of the neural model for these challenging real data sets. So in particular, this RAG1, RAG2 complex, we learned a lot from, and we can perform homogeneous reconstruction, again, showing RAG1, RAG2 complex in the pre-catalytic spliceosome. CryoDRGN 2 can reconstruct the structure similar to the reference. What we're really excited is the potential for ab initio heterogeneity. So here's kind of showing what we get with CryoDRGN 2 on the same pre-catalytic spliceosome data set. CryoDRGN BNB is this early branch and bound based procedure that failed to scale to real data sets. In the center, we have CryoDRGN with poses from the upstream homogeneous reconstruction. So this took 3.8 hours for training. And CryoDRGN 2 is completely ab initio without any poses assigned or any kind of heterogeneity assigned. And this takes less than 12 hours, also on one GPU. And just for another example, this is again, the RAG1, RAG2 complex. On the left is our with consensus poses, and on the right is ab initio, where we're fitting both the poses and the heterogeneity. And I think because of the kind of pseudosymmetry of this complex, maybe the ab initio case where we're fitting both poses gets the symmetry copy aligned correctly, and so there's less noise in the bottom part of this complex. And so the goal here is really to broaden the scope of single particle cryo-EM to more classes of heterogeneous biomolecular complexes that may not be able to get kind of accurate consensus reconstructions. So that's about all I have. So just to summarize what I've told you today, I've presented CryoDRGN, which performs heterogeneous reconstruction as unsupervised learning of this continuous distribution of protein structures from single particle cryo-EM images. We've contributed kind of this new neural representation for modeling high resolution cryo-EM density maps. And one of the main kind of features of this method is that this latent variable is this very general flexible framework for modeling heterogeneity. So with this model, we've been able to discover new structures that are missed in the traditional 3D classification, as well as visualize continuous dynamics. And then these two follow-up directions that I just mentioned are the automated classification and landscape visualization, and also CryoDRGN 2. And in the future, I'm very excited for kind of the intersection between machine learning for protein structures, for cryo reconstruction, to really enable new capabilities for how we can visualize biomolecular systems. So with that, thank you for listening. And again, big thank you to my advisors, Joey and Bonnie, my co-authors, Tristan and Adam, as well as many others in the Berger and Davis labs, as well as a lot of other people in the cryo community that have contributed to this work. So yeah, thank you.
Well, thank you very much, Ellen, for this fantastic overview, this really exciting method. So it's open for questions. I don't see any questions in the chat yet. Yeah, here is one. So it says, first of all, great work. How large is the range you can cover in terms of slow and fast movements? And how do you see potential for assessing the effects of protein interactions, and how complex in terms of sample complexity can you go? So there are several questions embedded here. The last one is, do you have plans to make us compare with other methods used for protein dynamics, for example, HDX data?
Yeah, I think there's an important point here, which is that when I say dynamics, I don't really mean kinetics. I mean kind of being able to visualize the maybe dynamical nature of these complexes from single particle cryo data, which is just a frozen snapshot. So we're kind of inferring the dynamics or the motions in a statistical sense by looking at the entire ensemble. And so, yeah, I think that's a very clear next step, is like, can we actually compare what you get out from single particle cryo-EM to the underlying dynamics or kinetics from other methods? So I think there's lots of other biophysical methods that you can use to measure some of these systems and really validate that, are these the motions that are happening? Yeah.
There's another question. So with CryoDRGN, can or should you impose symmetry? Still impose symmetry? Do you have to impose symmetry?
So you probably should not impose symmetry. The kind of power of this method is that, so we usually impose symmetry to get to high resolution, to kind of get more effective, to get more data by duplicating it. But I think one of the nice things is that, okay, our particles are not actually symmetric, perfectly symmetric. And especially in this RAG1, RAG2 example, maybe I forgot to mention, but the core is C2 symmetric, and the DNA extensions at the bottom are asymmetric. And so when you impose symmetry, you're actually just washing out what's going on in the heterogeneity, which is probably what you're interested in. So I would definitely say do not impose symmetry so that you can fit the underlying motions that are asymmetric.
Thanks. There is another question here. Do you have examples when CryoDRGN fails?
Yes. So, and then could you say a few words about limitations of the method when compared to other approaches? Yeah, definitely. The example that I showed, for some reason, I think I just put in the wrong slides because I wanted the text to explain it. But when I was talking about the interactive filtering of the data set, and there's actually all these weird edge artifacts and things like that. So that is some case that we've run into where instead of fitting the underlying heterogeneity, like the structural heterogeneity that you care about, the model is fitting these weird artifacts because they're heterogeneous and they're not modeled by the standard image formation model of just like rotation and projection, right? And so the model is like, okay, wait, there's this variability on the edge of the box. And so we're going to model that. And so that's interesting. Like in traditional approaches, you're doing this averaging technique, right? In 3D classification, you're just averaging for each class, you're just averaging all the images together. So you have outliers like that. You actually won't be able to see them, as assuming the outliers are relatively low relatively low frequency. But in CryoDRGN, those outliers are actually detected, and the model is very concerned about modeling them. And so in those cases, that can obfuscate the thing that you care about. And so our approach there is to perform multiple rounds of training, where you first train on the whole data set using a small model that is relatively cheap to run, to train, use that to filter these like large artifacts, and then run a higher resolution model on the filtered data set to really tease apart some of the smaller details or heterogeneity that is of interest.
Okay, there is, thank you, Alan. There's another question here. Can you comment on how well a CryoDRGN performs on proteins which can be considered rigid, such as it says only what a helix, moves or something like that?
So I guess there's maybe a clarification would be nice. Is it rigid where you just have like rigid bodies, or are we talking about small motions with just a single helix instead of a large conformational change?
Yeah, small motions with just a single helix. At least that's what's been described so far.
Yeah, so it's definitely harder in terms of just this statistical problem of learning, right? So if you have only a single helix moving, remember your images are very noisy, and so that's going to be a much smaller change in our images. So it's just a intrinsically harder problem to learn smaller motions. This RBD, or kind of COVID spike protein data set, the RBD is relatively small. So I think when dealing with very small motions, there's two things to consider. One is how large is the thing that's moving. If it's just a single helix, then the amount of density that changes is small, so that makes it harder. The other thing is how frequent is it? So is that motion present in a significant fraction of the data set? I think when it is in a lot of the data set, it's actually relatively easier to learn, or like easier to detect. When you have a very small motion that's also extremely rare, then that becomes even harder. And in theory, you could learn that, but there are no guarantees. Yeah.
Now, another question, is CryoDRGN compatible with particles picked using RAP-EM?
Yes, I think, yes, it should be. The inputs to CryoDRGN, I think we support parsing Relion and CryoSpark inputs, but I'm assuming the format for warp is also like the standard star files from that Relion uses. But if it's not, you can definitely file a GitHub issue for a feature request.
Thank you.
There's another longer questions. How resistant is CryoDRGN to noise? And it goes on, imagine a membrane embedded protein, which in a typical reconstruction will still have strong micelle density, and the micelle itself is most likely heterogeneous in shape and size. If there are dynamic motions in the membrane domain of the protein, can the program sample and filter them from noise?
Yeah, so this is another case of maybe where CryoDRGN fails, which is where you have these membrane embedded proteins and the membrane itself is very heterogeneous. The model is modeling kind of the motions there, kind of the changes in all of the lipids there. So there's kind of two strategies. And I should also say that this is totally just anecdotal or empirical. We have cases where it doesn't work because the micelle is really large and the actual transmembrane domain is very small. And so it's just harder. In other cases, like VHPases, the membrane is like the whole complex is like pretty large as well. And so there we haven't had issues with dealing with micelle heterogeneity. So the actual kind of one approach is to do signal subtraction. So if you can, in the consensus reconstruction, draw the mask around the particle and draw the mask around the micelle, you can computationally delete the micelle, and that just significantly reduces the amount of density constituting the micelle and perhaps guides the model towards modeling the heterogeneity of the system. So that's one thing that we recommended.
Okay, that's Anastasia. You can maybe say your questions loud.
Yeah. Hi, thank you, Ellen. I actually had two questions. So I was wondering what your latent space analysis can tell about the likelihood of certain states. So if you have two samples, can you in principle compare the population distributions of those two samples?
Yeah, so that's definitely something that we're interested in. And this landscape analysis tool kind of facilitates that because it's assigning states and then quantifying kind of the number of particles associated in each of those states. I think the key kind of question there, or maybe problem there, research problem there, is how do you define state? Which is, I think, guided by the practitioner, the structural biologist, is like, what is a distinct state? What makes one state distinct from another? It's interesting because you're kind of imposing a discrete model on the ensemble. And you're saying, okay, we're not like, we've learned this continuous model, which is great because we can model like the complex nature of our system, but actually we want to just summarize it in terms of a few discrete states. And then it becomes maybe a expert guided question of like, where do you call something a state? And once you've made that definition, then you can quantify the number of particles associated with each of those states. So that's kind of the way I think about it.
I had a question also to find out, how much training or retraining do you need when you have a new data set? Do you have, can you use a model that you already derived, from other data from another system to apply to a new one?
Yeah, so this is actually, the short answer is no. And this is actually kind of a common confusion because a lot of machine learning models are trained across like a large data set or lots of a huge data set and then applied in like new settings. Here, we're doing kind of training each new data set we get, we train a new model. So it's kind of the same as doing reconstruction. Like there's no way you would reuse the reconstruction from one data set for another.
So how much information do you need to, to start the training? In other words, how much reconstruction data, reconstruction data do you need for that?
So the inputs are just the single particle images and poses, not for CryoDRGN 2, but it's just the extracted single particles. And then the success of reconstruction is kind of based on, it definitely abides by like garbage in, garbage out. So if the traditional 3D reconstruction approaches work, then probably your data set is kind of like your particles are sufficiently high quality to also train CryoDRGN. If the traditional reconstruction doesn't work because of heterogeneity, then that's like a better reason to try CryoDRGN. But if the traditional reconstruction doesn't work because your CTF parameters are wrong, or you have a lot of denatured particles that are just totally broken, then you're also not really going to get anything interesting out of the system either. But yeah, just conceptualizing this model, even though it's this machine learning system, conceptualizing it as just another reconstruction software, I think is useful for understanding how it and when it should be applied.
Thank you.
Maybe I have one last question. So if you, so you showed us, so you, in one of your example that you, so one subpopulation or like subspace of the latent space, you constructed it with CryoDRGN and with traditional reconstruction, and it looked like CryoDRGN could reconstruct much more details than traditional, the traditional approach. So
yeah,
is that because it uses kind of implicitly the information from the entire space? That's
yeah. So I think you're referring to this, and the short answer is yes. So and that kind of makes it interesting. That raises questions of validation, but because we're training the model on the whole data set, you can see that the bottom part of this complex is higher resolution, or maybe like matches the resolution is all the class C particles. And maybe this additional helix here is lower resolution. can maybe borrow additional signal from the class E particles, perhaps. But if you just isolate only the images that contribute to C4, then you'll get this lower, like 7.6 angstrom resolution structure. Maybe that should say you can only trust the results of CryoDRGN up to 7.6 angstroms, perhaps like any specific details maybe you need. If you really care about one specific state, you want to validate the specific state in high resolution, you really just need a lot of images associated with that, or particles associated with that state. I think it's interesting because instead of being kind of confirmatory of the 3D model of your system, now it's more like hypothesis generation of, okay, maybe we have like we have a structure here. How do we use other methods to validate the structure? So I don't know.
Thanks.
There's one more question here. Everybody's really interested. You mentioned previously filtering out particles with artifacts coming from recentering to make training more robust. Is re-extracting centered particles something you recommend in general?
The artifacts in particular were because the particles were recentered when they were before they were deposited to Empire, or actually before they were transferred to us. So I'm not totally sure why they were recentered. I mean, that is just a common thing that one does when you process cryo-EM data sets, is you recenter it just because the search space of the poses is like typically set to like plus or minus some percentage of the box. So you just recenter it so you can maybe more accurately estimate the shifts. In this case, so the data set was already pre-centered, and because of that, and maybe not re-extracted, there's this weird edge artifact. And so I guess in general, it should be good to re-extract the particles so you get the whole thing and you don't have to deal with some of these edge artifacts.
So Ellen, how complicated, or what is the learning curve for implementing or using your systems in a new place, like in our center in the VB, for example? What would be the learning curve to do that?
Yeah, so I've tried to describe every step of how I process the data sets in the tutorial. I think there's some learning curve. I think the learning curve for actually for understanding how to run the system is pretty low. I mean, it's just a command line tool. And there's like default settings. So hopefully that's like relatively straightforward. The actual learning curve, I think, is how do you analyze the models, which I think is, so there's definitely like some Python in the Jupyter notebooks. There's a lot that we've tried to automate with kind of this like CryoDRGN analyze pipeline and the new landscape analysis pipeline. But there definitely is a learning curve in just how do you interpret the method? Like how do you interpret this continuous latent space? And a lot of it is kind of up to it's not up to the algorithm. It's definitely up to the researcher of what do you want to, like what insight do you want to maybe validate from your single particle cryo-EM. So I think the learning curve is both a learning curve in using the tool, but also in what do you want to get out of single particle cryo-EM.
Mm-hmm. Yeah, thank you very much. That sums it very well. Okay, I think someone else has another last question, and that's if you have a few more minutes.
Mm-hmm.
So someone is a French master student, and I'm currently doing a 2-month internship here with a team. I had sent you an email to find out if it would be possible to go to an internship with your team because I find your work fascinating. So I had no response to my request. So it's time to try my luck again.
I'm sorry for missing. So definitely, there's a lot of emails and I definitely miss things. So definitely feel free to like re-email me, especially when I'm traveling. So that's when I like miss all the emails. I'm sure a lot of people can relate to that. The specific kind of question. So I'm in transitioning right now. So I don't really have a team to offer any like side projects or anything like that, but I'm also happy to chat and discuss just science and cryo-EM. Yeah.
So one last question, a scientific question, that's about your number of latent variables. Why is it so large? And you know whether. Okay, go ahead.
Yeah. So the I guess just to clarify, each particle is associated with an embedding, a latent embedding. So each image has like a assigned kind of latent variable value. And that's kind of one of the features of the method, is that we have this encoder that can encode, like once it's been trained, it can encode each image to some location in latent space. So there's one per image, so it just depends on the size of your data set. And then together you can look at that, ensemble, and maybe that tells you something interesting. The dimension of the latent variable is typically rather or low-ish, low dimensional. So the dimensionality of the input images is n by n, which is pretty high, especially for large images. The dimension of the latent space is typically 8. I think that is kind of the default dimension that we've converged to as both being expressive enough to learn these complex distributions, but also still being low enough to be interpretable and kind of train accurately.
Well, wonderful. Thank you very, very much for this nice discussion, Ellen, and for the wonderful overview of your work. Now, and I think we have to wish you best of luck and continued work, you know, to integrate this method with structure prediction and other exciting, you know, exciting goals.
Yep. Thank you so much.
Thank you.
Thank you.
So goodbye. Goodbye, everybody.
Bye, everyone.
Thanks for everybody for joining. Goodbye.